All right, here's a continuation of the notes, just starting with passive transport and facilitated diffusion. So materials can also move through some proteins passively from a high concentration to a low concentration. So I think that's one of the biggest things to remember with passive transport is we're always moving down the concentration gradient. And what will be used in facilitated diffusion is protein channels. So protein channels must be stimulated to open, and it's usually specific for the particles. So larger particles and ions can move this way. So it's like a key almost. It's just that it doesn't take any energy. So you guys have this diagram in your notes, and I just really like it. So it just shows you a molecule on the outside of the cell binds to what we call a carrier protein, and it basically just will open it, almost like a turnstile maybe kind of thing, and it will open it, and the protein will transport that molecule through that cell membrane, and it will be able to go out the other side. So facilitated diffusion is just kind of going through a turnstile. It's just that turnstile for the cell membrane is very specific to the thing that's trying to pass through. So that's facilitated diffusion. Here's another good example. So ion channels allow certain ions to pass through the cell membrane, because if you remember earlier, charged particles cannot pass through that cell membrane, and ions are charged. So what we have are sodium and potassium channels, and so what will happen is that it will get opened by a specific either sodium or chlorine or potassium, and it will open, and the gate will be able to, like just for sodium will be able to pass, whereas, for example, chlorine would not be able to use this channel. So again, it's very specific to the substance. So I recommend that you go through these questions, because we went through these in class, and review through your passive transport. If you're wondering what you can write on the left-hand side of your Cornell notes, this would be a great opportunity to kind of go try to find where these would fit, so that you can definitely review. So I would go through your notes and review these questions. So now we're on to active transport. So active transport requires ATP, and it usually moves materials against the gradient, or it moves them in bulk. It can use protein pumps, or they can utilize vesicles. So remember, vesicles are created in the Golgi apparatus. Vesicles out is called exocytosis, so exo and exit kind of go together. And vesicles in is endocytosis, so endo means in. There's two types of endocytosis: phagocytosis, which is food particles or bacteria coming in, and pinocytosis, which is liquids coming in. So there's a lot of vocab words on that slide that you definitely need to know, so it's probably worth making sure you're starring, you're underlining those, and that you're making sure that you know what those are. So protein pumps, so you have this example on your notes as well. So basically what will happen is the sodium-potassium pump actively transports sodium ions and potassium ions against their concentration gradient. So 3 sodium ions and a phosphate group from an ATP bind to the pump. Okay, so this is just showing you that it takes energy when it's talking about that ATP. Okay, and as soon as that ATP is there, what will happen is those 3 sodium ions will be able to go outside of the cell. Vice versa, the potassium ions will then enter the cell, and it will be able to activate it and move inside the cell. So this is really important when we're talking about muscle twitches and how muscles work. The sodium-potassium pump is a really important protein pump when dealing with like muscle control and how muscles fire, etc. So exocytosis, again, these vesicles are coming from the Golgi apparatus. So this is a membrane-bound vesicle, and it just simply will kind of merge with that cell membrane and then release out what was inside the vesicle. So this could also be from lysosomes as well, because they might be exiting trash from the cell, if you will. Endocytosis, so this could be from like phagocytosis, remember, is with food, or pinocytosis, so it's just basically bringing in things from the outside, creating a vesicle, and then having that pinch inside the cell. So there's the difference between endo and exocytosis. So our active transport review, again, we'll have done this in class, so it's probably worth going through and making sure that you can answer those questions. Those are a good review for you for the notes and the material. And then just kind of going further, with the cell membranes, proteins have more than just the function of transporting materials. So there's 3 other things that the proteins can do. So proteins are marker proteins, so it helps identify what type of cell it is, and this enables a person's immune system to identify its own cells from invading cells, which is really important. And that's also why it's hard to fight some of the viruses that we have, because some of those viruses kind of pretend like they're our own cells, so it's hard for our immune system to go find them. Okay, proteins are obviously enzymes, which again help speed up chemical reactions and assist in metabolic processes within the body. And then enzymes are also receptors, so these are proteins that allow the cell to interact or communicate with other cells. And so you guys have this diagram as well, where we're looking at proteins, especially like hormones, for example, interacting with other organisms inside and outside the cell. So there's your notes on proteins in the cell membrane, so membranes, and transport, active and passive transport.